My name is Desiree Santoyo, and it is the 9th of February 2024. First, we're going to start talking about testosterone, which is a lipid macromolecule. The Leydig cells found in the testicles and ovaries are known for producing testosterone. It starts off by the hypothalamus generating a hormone called GnRH, which stimulates the pituitary gland to produce LH, or luteinizing hormone. LH and the G protein bind to LH receptors on the Leydig cells, stimulating CAMP production. CAMP stimulates cholesterol translocation from intracellular sources to the mitochondria. This is known as the rate-limiting step to producing steroids. The mitochondria then forms pregnenolone. Pregnenolone is modified and converted by the smooth ER into testosterone. Testosterone is then secreted out of the plasma membrane into the bloodstream. Testosterone has many targets, and specifically these are for biological effects. These targets differ based on age and sex of the individual. Now, moving on to a secreted protein, we're going to be talking about insulin. Insulin is produced in beta cells within the pancreas and is also stored within the pancreas. It starts off by the nucleus sending mRNA to ribosomes on the rough endoplasmic reticulum, or ER, to produce insulin. Ribosomes then produce proinsulin, which is then further modified by the rough ER to proinsulin. Proinsulin then goes through the Golgi apparatus, through the cis phase, and comes out through the trans phase, and is matured as insulin to be stored in the beta cells. Insulin is then stored until this process takes place. Glucose from a meal goes through the GLUT2 channel on the beta cell. Once glucose enters the beta cell, the glucose undergoes glycolysis and goes through the Krebs cycle producing ATP. ATP binds to the receptor of the potassium channel. This prevents any potassium from exiting the cell, causing an influx of potassium in the cell. This leads to depolarization, causing calcium to increase. Calcium binds to the receptor of insulin, causing exocytosis, meaning exiting the cell and going into the bloodstream, seen here, which is exocytosis. Insulin then travels to other cells throughout the body to bind to receptors and allow glucose to enter the cells. This is an important effect because if insulin cannot bind to those receptors, it will lead to high blood sugar, which is hyperglycemia, which can lead to death if not controlled. Lastly, we're going to be talking about an intracellular protein called actin. Actin is a globular protein that is very prominent in eukaryotic cells. The nucleus sends mRNA to ribosomes for protein synthesis. Actin is an intracellular protein, specifically a cytoskeletal protein, meaning it is synthesized by free ribosomes. Once synthesized, the actin goes to the Golgi apparatus to be stored and properly distributed. Actin is very important for the maintenance of the cell structure and influences the motility of the cell. So, actin is often distributed to the cytoskeleton of the cell. However, actin plays an important role in muscle contraction. Movement is not feasible without actin because of the fact that actin binds with myosin to produce muscle contraction. As you can see, macromolecules differ in several ways, however, they have very similar pathways in how they function.